Welcome back to this next video in the series of lectures on the cell cycle. In the last video, I've told you that after the activation of the mCDK, one of its important function was the polymerization of the microtubules. Once you have gone for the polymerization of the microtubules and you have dissolved the nuclear envelope from the centrosome, from both the poles where you have got the centrosome, three kinds of the microtubules they are originating. The first type of the microtubule, they were the kinetochore microtubules, and they were actually interacting with the kinetochores that were attached to the sister chromatids. The second important microtubule, they were the astral microtubules, and they were actually interacting with the cell membrane on both sides. The astral microtubule originating from this centrosome, they were interacting with this part of the cell membrane. The astral microtubule originating from this centrosome, they were actually interacting with this part of the centrosome. And the third type of the microtubule that were originating, that were the polar microtubules. The kinetochore microtubule, they were directly interacting with the sister chromatids, while the astral and the polar microtubules, they were not interacting with the sister chromatids. The polar microtubule, they were interacting with each other, and the astral microtubules, they were actually interacting with the cell membrane. Once you have attached the microtubules to all of the pair of the sister chromatids, this is actually what you see, that all of the pairs of the sister chromatids that align in the middle of the cell, which we call is the equatorial plate. And I've told you that the bipolar attachment of all of the pairs of the sister chromatid, that is necessary, that is mandatory if you want to move from the metaphase into the anaphase of the cell cycle. And this bipolar attachment, that was actually ensured by a particular checkpoint which was known as the spindle assembly checkpoint. Now let us discuss this spindle assembly checkpoint in a little bit detail, how this spindle assembly checkpoint is actually ensuring that all of the pairs of the sister chromatids, they have got bipolar attachment. The key here is the tension on the kinetochores. If you remember, I've told you that during the S phase of the cell cycle, when the DNA gets replicated, all of the pair of the sister chromatids, they are attached to each other by a set of the cohesion protein. Like if you talk about this pair of the sister chromatids, both the members of this pair are attached to each other by the cohesions. Both of these sister chromatids, they are attached to each other by the cohesion. And similarly, this pair of the sister chromatids, they are attached to each other by the cohesion protein. Now, when the microtubules, they get attached to these sister chromatids, and if they want to pull the sister chromatids to the opposite pole, like if you see in this member of this particular pair, it is attached to this kinetochore microtubule from this pole, and this member of this pair is attached to the kinetochore microtubule from this centrosome. Now, if the microtubules, if they are pulling these two sister chromatids in the opposite direction, the problem is that is these two members are attached to each other by the cohesion proteins, a tension is created on the kinetochores, because these kinetochore microtubule, they are attached to the kinetochore on this sister chromatid. This kinetochore microtubule is attached on the kinetochore on this particular sister chromatid. So if you are pulling them and if they are attached to each other, there is a tension created on the kinetochores. Now, if this tension is felt on all of the pairs of the sister chromatids, I am emphasizing on this, on all of the pairs of the sister chromatids. If this tension is there, that means that everything is okay. You can move into the anaphase of the cell cycle. But if this tension is not felt on even a single pair of the sister chromatids, that means that the things, they are not right, and you cannot allow the cell to move from the metaphase into the anaphase. If you allow the cell to move from the metaphase to the anaphase when there is no tension, say for example on this pair of the sister chromatids, that is going to create a problem. For example, if you talk about this particular pair of the sister chromatids that is attached to the kinetochore microtubule from only this particular pole, the other member of this pair is not attached to the kinetochore microtubule from this particular centromere. So when the cell divide, like if these two sister chromatids, they are separated from each other, this one sister chromatid is going to this pole, the other one is going to this pole. if you look at this particular scenario that I have drawn over here, at the end of the cell division, this particular cell is going to have 1, 2, 3 and 4 chromatids. The other cell that is going to have only 1 and 2 chromatids. So this particular cell after division will have 2 chromatids. This particular cell after division will have 4 chromatids. Both of the cell, they will be abnormal, and if they are abnormal, that means that your cell, they can be aneuploid in nature, and that is one of the possible reason for the cancer cells to arise. Now, how the spindle assembly checkpoint is going to ensure this particular tension on all of the kinetochores? Let us discuss this in a little bit detail. I have reproduced the sister chromatid pair again over here, that this pair of the sister chromatid, one of the member is attached to the kinetochore microtubule from this pole, but the other member of this pair is not attached to the kinetochore microtubule from this particular end. We call this as the unattached kinetochore. Unattached in the sense that to this particular kinetochore, no kinetochore microtubule is attached. So when there is no kinetochore microtubule attached to this particular kinetochore, that is actually providing a binding site for the proteins that are present in the spindle assembly checkpoint. One of the important protein that is present in the spindle assembly checkpoint that interacts with this unattached kinetochore is the MAD1 protein. Now this M is for mitotic, this A is for arrest, and this D is for deficient. This is known as the mitotic arrest deficient protein, because in mutant cell, when there is a mutation in this particular protein, actually the process of the mitosis cannot be arrested when there is a problem. Therefore, we call this as the mitotic arrest deficient protein. And specifically this MAD1 protein that is going to interact with the unattached kinetochore. Now, if you look at this unattached kinetochore over here, 2 MAD1 proteins, they are going to attach over here. So these 2 MAD1 protein, they are actually going to recruit another MAD protein, which is known as the MAD2 protein, to the unattached kinetochore. Now let us discuss this MAD2 first. This MAD2 protein, it actually exists in 2 forms. Its first form is known as the O form, O for the open form. So when the MAD2, it is in the open form, or when it is in the O form, it is actually inactive in nature. The same OMAD2, when that is converted into the CMAD2, the C is for catalytic, this CMAD2 form of this particular protein is the active form of this protein. Now, when the MAD1 that attaches to the unattached kinetochore, it actually recruits the inactive OMAD2 form of this particular protein. When the OMAD2 that interacts with the MAD1, this OMAD is converted into the CMAD. That means the interaction of the MAD1 is actually activating the MAD2 protein by converting it from the O into the C form. Now a complex is made that contain 2 MAD1 protein and 2 CMAD2 proteins. Now this particular complex containing 2 MAD1 and 2 CMAD2, they are actually going for the conversion of all of the MAD2 available in the cell into the CMAD2. Now that means all of the inactive form of the MAD2 protein is going to be converted into the active form. Now when the CMAD2 that is activated, this CMAD2 is actually going to stop the cell cycle at the metaphase when there is even a single unattached kinetochore. Now how this CMAD2 is going to achieve this? This CMAD2 is going to interact with a particular protein known as the CDC20 protein. And this CDC20 protein is very important if you want to move from the metaphase into the anaphase of the cell cycle. When the CMAD2 that interacts with the CDC20, the CDC20 actually is in the inactive form. When the CDC20 is in the inactive form, it cannot actually go for the activation of a protein complex which is known as the anaphase promoting complex. Now, as the name indicates, this is anaphase promoting complex, and that means if this is in the active form, that is actually going to promote the anaphase. But if the anaphase promoting complex is in the inactive form, that means you cannot move from the metaphase into the anaphase of the cell cycle. This is how the unattached kinetochore signals that the cell cycle should be arrested in the metaphase, and it should not be allowed to move into the anaphase, because if you allow this, you are going to have abnormal cells, and this abnormal cell can create a problem in the body. Now, what are the functions of the anaphase promoting complex? We will discuss that in the next video.